Welcome back. This is day 5 of genetics. So today we're going to be talking about how do DNA and RNA assist in protein synthesis. Well, yesterday I told you guys that protein synthesis can actually be broken down into 2 steps. Yesterday we talked about transcription, right? We saw that DNA was copied into mRNA. And today we're going to be talking about the second stage of protein synthesis called translation. Now, this is a word I'm sure you guys have heard before. In fact, I'm sure you're very familiar with Google Translate while working on your Spanish and French homework. So when we translate something, we're interpreting it in one language and translating it into another, or changing it into a different language. So today, the languages we're going to be dealing with are first the language of mRNA, and we're going to be translating that or interpreting it in an amino acid sequence that's then going to be used to create proteins. So remember that when we're talking about protein synthesis, we're ultimately trying to create proteins. There's just a couple of different steps that we have to do correctly in order for that to happen. So before we get into that, I want you guys to pause this video, answer your 2 do now questions, and press play when you're ready to review it. So number 1, we've got just a slight math problem. I'm sure you guys can handle it. We're told that if 15% of a DNA sample is made up of thymine, what percentage of the sample is made up of cytosine? So remember that because this is a DNA sample, we're going to be dealing with the 4 bases A, T, C, and G. A always pairs with T, and G always pairs with C. So if 15% of our sample is thymine, because A and T are always paired together, that also means that 15% of our sample is adenine, okay? But then if we add up both of those numbers, 15 plus 15 equals 30. So that means we now have 30% of our DNA sample accounted for. But we also know that all 4 of these bases have to add up to be 100% of our DNA sample. So what do we have to do next? Well, we have to take this 30% and subtract it from 100. So 100 minus 30 is going to be 70%, but that's not our final answer. So we're not done yet. We still have to account for our samples of G and our samples of C, which means we're going to divide this number 70 by 2. That gives us a final answer of 35%, which means our sample is made up of 35% G and 35% C. All right. Number 2, chromosomes can be best described as what? So we typically see chromosomes drawn as these tightly coiled structures. We usually see them represented as an X shape, okay? And the reason they're tightly coiled is because that DNA is bound up really, really tightly inside of them. So the answer here is going to be D. Coiled strands of genetic material, or DNA, is what make up those chromosomes. All right, let's get to it. So the very first thing we're going to do today is read a short passage about translation and what it is. So as always, we're going to read through this passage together, highlight or annotate anything you think is important, and we'll use that information to answer questions 1 through 3. All right, from RNA to protein. So far, the genetic information stored as base sequences has moved from the nucleus to the cytoplasm by using RNA. Another problem remains. How do we use the nucleotide base sequence in the RNA to build a protein with the correct sequence of amino acids? This problem involves a change of language from the base sequence language of RNA into the amino acid language of proteins. This process is called translation, and it occurs at the ribosomes. Built into every living cell in the world is a genetic code. It is called the triplet code. Each different combination of 3 bases makes up a word called a codon. Each codon represents a specific amino acid. Each of the 20 amino acids has at least 1 codon, and most have more than 1. This genetic code is universal. In other words, all organisms on Earth use the same genetic code. For example, the codon GCA stands for the amino acid alanine in all life forms, from bacteria to trees to human beings. This similarity among living things is good evidence that all organisms evolved from a common ancestral life form in Earth's distant past. So now, using that information, pause this video, answer questions 1 through 3, and press play when you're ready to review it. So what is translation in terms of protein synthesis? Well, remember when we're translating something, we're interpreting it between different languages. So today we're going to be changing the language of RNA into the amino acid language of proteins. Number two, what is a codon? We've heard this word a few times by now. And so hopefully we know that codon is a combination of 3 bases that codes for a specific amino acid. And remember those amino acids are going to be combined together to form proteins. And number 3, how is the genetic code universal? Well, we describe it as universal because all living organisms on Earth use this same mRNA code. And as the passage said, this is actually some pretty good evidence that we all came from a common ancestor at some point, because every single organism on Earth uses this same language, which is pretty cool if you think about it. All right, so now let's go through some animations trying to figure out how does mRNA actually code for proteins? How does translation actually work? So by now we know that mRNA has to leave the nucleus and it's going to go to the ribosomes in the cytoplasm, right? It's going to take that copy of DNA over to the ribosomes to make proteins, because proteins are built from those instructions that we find on mRNA. So here we've got that mRNA sequence, and eventually it's going to be used to form that amino acid sequence. But how does that happen? Let's find out. Right, so here we've got a DNA sequence. Yesterday we talked about the process that was needed to take this DNA sequence and turn it into an mRNA sequence, okay? This was called transcription. We're making a copy of that DNA, and that mRNA strand is going to take that copy, leave the nucleus, and take it over to the ribosomes. That ribosome is then going to read that mRNA strand and start churning out an amino acid sequence. Okay, but how do we get proteins from these amino acids? Like, there's a lot of gray area between these last steps. So how does it happen? And how can we code for 20 amino acids when we only have 4 DNA bases? So yesterday we learned that there are 20 amino acids, but we only have A's, U's, G's, and C's. So how does that happen? Well, we need to know that mRNA codes for proteins in triplets, and those triplets are called codons, right? So when that ribosome reads the mRNA sequence, it's actually reading it in chunks of 3. It's going to read it according to those codons. And so what ends up happening is each of those codons, each of those series of 3 bases, is going to correspond to 1 unique amino acid. So here you can see that this chunk right here, this codon codes for the amino acid methionine. This amino acid is going to call for ARG, VAL, and the rest of these that we'll talk about in just a second. But each of those codons codes for 1 specific amino acid. Right, so depending on the order that those letters are arranged in, means that it's going to code for a different amino acid sequence. Oh, there's another one. So a codon, in case you didn't know by now, I forgot I included this as well, is a block of 3 mRNA bases that is going to code for an amino acid. Right, now this chart I know looks super complicated, but I promise we can figure it out. So this chart is actually showing us or outlining the mRNA code. And I will tell you guys how to read this. I think I actually might wait until class tomorrow, so don't panic just yet. So this mRNA sequence, as I said, is actually used for all life. Okay, every single living organism on Earth uses this mRNA code. And so as we said earlier, this is the strongest support for a common origin or common ancestor for all living things on Earth. We all use the same genetic code. And yesterday in my, I think, 3rd period class, someone brought up the point, well, what happens if DNA polymerase or RNA polymerase makes a mistake when they're creating these duplicate strands? And your body actually has insurance for that. It knows that there might be a possibility of mutations, and so it protects you from danger. And the reason, or the way that it does this, is that several codons actually code for each amino acid. So if you look at this chart, we see that the sequence UUU and UUC both code for the same amino acid. So if there's a mistake where a U is actually inserted instead of a C, it's not a big deal because they actually code for the same amino acid. The same thing happens here, where all 4 of these sequences code for serine, the same amino acid. So regardless of if there's a mistake at this last letter, the same amino acid is still coded for. So the reason it does this is to protect you against mutations, meaning that if there accidentally is a mistake, it won't affect you in any sort of detrimental way, because they all code for that same amino acid. Okay. Now, I won't ask you to memorize every single amino acid. That's insane. But I do want you guys to be familiar with just a few. And the first ones I want you to know are the start codons. When the ribosome is going to read that mRNA sequence, it knows where to start because it sees or recognizes this one sequence, AUG. So this AUG is actually right here on our chart, and it codes for the amino acid methionine. And whenever the ribosome sees that codon AUG, it knows, okay, this is my cue, this is my time to start reading this sequence. All right, and we also need to be familiar with these stop codons. Similar to that stop codon, this is a cue for the ribosome to stop reading that mRNA sequence. And there are actually 3 different stop codons that we need to know. So the 3 stop codons are UAA, UAG, and UGA. Anytime the ribosome sees one of those stop codons, it knows it is time to stop reading that mRNA sequence and move on to the next one. So again, we do not need to know every single one of these amino acids, but we do need to be familiar with these stop codons and start codons, because in just a few days, we'll be actually coding some sequences ourselves. And so we need to know that AUG is our cue to start, and UGA, UAA, and UAG are our codes to, or our cues to stop reading that mRNA sequence. Right, but again, I will actually show you guys how to read through this mRNA chart in class tomorrow, but I'm not gonna worry about it on this video. I should not have gone back. All right, so now how are these codons actually matched to an amino acid? Well, we've got another character to introduce, and this is our good old friend called tRNA. You can consider him like a distant cousin of mRNA. They're both ribonucleic acid, but they do different jobs. They're a little bit separated. So what tRNA does is this is actually called transfer RNA. And it is going to transfer the amino acids specifically to that mRNA sequence to create the amino acids. Now, that was not a good sentence. That did not make sense. But let me show you what this looks like in terms of this. I'm crumbling, guys. I haven't had enough coffee. Let me show you what this looks like in our, oh my God, I can't think of the word. Visual, graphic. Sure, we'll go with it. So tRNA is actually shaped like a little T, and it kind of carries a backpack with it. So this circular amino acid, I like to think of a little backpack that it's carrying around and then it's going to drop off. So what happens is the tRNA is actually carrying a codon sequence that's opposite to the codon sequence we see on the mRNA. And because of this, we actually call it an anti-codon. It is opposite to the codon sequence. And so tRNA knows to match up with the codon according to its anti-codon. I'm going to write that it's the opposite of the codon. So here we see that the codon on mRNA is AUG. The opposite of that is UAC. So this is a tRNA that knows, hey, this is my cue, I gotta go. It's time for me to drop off my amino acid. And then this other tRNA is going to come in and do the same. And soon enough, all of these amino acids are going to start to hook together because that tRNA is actually gonna leave. He's like, I've dropped off my amino acid, I'm good to get out of here. It's like that SpongeBob meme, all right, I'm gonna head out, okay? He drops off his amino acid and then he's good to leave. But the amino acids start hooking up together and linking. And we know that when we have a chain of amino acids, we're actually going to form a protein. Right, so I actually do want you guys to write down that an anticodon is a block of 3 tRNA bases, and it is the opposite of the mRNA codon. All right, so this is the process of translation where we're turning that mRNA sequence into protein. So the working instructions that the cell is following is mRNA. Remember, this is the copy of DNA that is being used to create proteins. Here's that sequence of mRNA. The reader is actually the ribosome. The ribosome is going to read that mRNA strand and use that information to bring in the correct tRNA sequences. So that means that the transporter is known as transfer RNA. This is what's actually carrying the amino acids. Right, and it's going to drop them off. To form proteins. And I think I have a visual of that. Yes. So again, the tRNA is going to match up with its correct codon according to that anticodon that it has. Right? And it's going to drop off the amino acid backpack that it's carrying. So it's going to take off that backpack, and all of the tRNA molecules are going to do the same for each of their codons, until eventually we're left with a complete chain of amino acids. Here it comes. I knew I had some animations there. Until we have a chain of amino acids. Oh man, I'm really struggling with PowerPoints these days, guys. Let me try that one more time. Come on, come on. There we go. And as these tRNAs drop off their amino acids, they're going to link together to form a protein. All right, so let's look at this from start to finish, from gene to protein. We know that DNA, as we found out yesterday, is contained within the nucleus. It has to stay there because I dropped my pen. We have to keep it protected inside the nucleus. It cannot leave. So we still need to get those genetic instructions out to the ribosomes though. So that's where we bring in the mRNA to make a copy of that DNA and take it over to the ribosomes. So here we have that sequence of mRNA. This process is known as transcription, but we're not done yet because we still have to make that protein, and that involves a little bit of translation. So that brings in the next stage where we're using mRNA to make proteins. The ribosome reads that mRNA sequence, brings in the correct tRNA structures that are then going to drop off their amino acids and synthesize a protein. Whoa, that was a lot of information. So let's look at that one more time with a different visual. So this is the same graphic we used yesterday, right? We know that DNA is locked inside the nucleus. During transcription, we're going to make a copy of that mRNA or of the DNA sequence using mRNA. mRNA is single-stranded, so it's able to leave the nucleus. It's going to take that information over to the ribosomes, where the protein factory is. That's where the proteins are made and are synthesized. But we're still dealing with a few different languages here. We've got the language of mRNA and we've got the language of amino acids. So that we then have to bring in another helper in order to undergo translation. This is where tRNA is going to come in and help. So you can kind of think of this as the translator. Okay, this is transfer RNA because it's going to transfer the amino acids. So tRNA comes in, it's going to bring in, it's a truck, it's going to dump over its amino acids, and that's going to happen for every single codon that's being read. Okay, that happens over and over and over until we synthesize all of these amino acids together as a chain to form a protein. And proteins are what make up your traits. So the reason you have your color hair and your color eyes are all due to proteins being arranged in a certain and specific way, and that makes up your physical traits or your characteristics. All right, and that is all I've got for you guys today. So now I've got one more thing that I want you to do, and that is I want you to make sense of everything we've talked about. So everything we talked about yesterday with transcription, everything we talked about today with translation, I want you to put it in your own words. So on your worksheet, there is space at the bottom for you to kind of write a short paragraph using a few of the vocabulary words that we have talked about, and put this process of protein synthesis in your own words. So I will be checking those, make sure you complete that passage on your own. But other than that, guys, all you need to do is fill out your exit ticket, submit your notes, and have a wonderful afternoon. I'll see you guys tomorrow.